All right. I'm going to go ahead and get started here with the housekeeping before we get the talk underway. So first of all, welcome to everybody. This is our first of our Science of COVID-19 mini-series as part of our regular seminar course. Just a quick refresher for the students that are in the class and also those that are joining us for the first time today. We generally encourage you to keep your camera off and your microphone muted during the talk, and then during the Q&A session, we would love it if you have questions, if you could either put your question in the chat to be read out by me, or you can use the raise hand function, and then I will call on you and you can turn your video on and unmute and ask your question yourself. And then just a quick note for the students that are either in the MolES 520 or the Chem 597 course, please remember that the links for your attendance will be put into the chat, so be sure to follow those to get credit. And then we can go ahead and get started here. So I'm very excited to introduce our first speaker of the Science of COVID-19 mini-series that we're doing, who is Dr. Keith Jerome. And he is a physician and medical researcher on the faculty at the University of Washington and at the Fred Hutchinson Cancer Research Center. Dr. Jerome leads the Virology Division within the Department of Laboratory Medicine and Pathology at UW, also known as UW Virology. Under his guidance, UW Virology designed and implemented molecular testing assays for a wide variety of human viruses, including SARS CoV-2, hepatitis B and C, enterovirus, BK virus, and cytomegalovirus. The program provides diagnostic support for stem cell transplant and other patients in the Pacific Northwest and throughout the country through its reference testing services. UW Virology has been a leader in the national response to COVID-19, providing clinical testing and support for research studies evaluating new therapies and vaccine candidates. In addition to his clinical efforts, at Fred Hutch, Dr. Jerome leads a research group focused on the biology of chronic viral infections. He has pioneered the use of gene editing and gene therapy as potentially curative therapies for HIV, hepatitis B, human papillomavirus, and the herpes virus infections. He currently serves as co-principal investigator and NIH contact investigator for Defeat HIV, Fred Hutch's Martin Delaney Collaboratory, working towards a cure for the HIV disease. Dr. Jerome earned a Bachelor of Science from Georgetown College before earning a PhD in microbiology and immunology and his MD from Duke University. So with that, let's please welcome Dr. Keith Jerome.
All right, thanks very much. It's a pleasure to get to speak, so I appreciate the invitation. I'm going to share my screen here, and you can, all right. Can you see the slides?
Yep, looks good.
Corin? All right, perfect, great. Yes, I didn't realize I was actually kicking off the mini series of COVID talks, so that's exciting. So I want to take about 45 minutes and sort of give you a very high-level overview of what we've been doing for the past year in terms of COVID testing and supporting research and supporting efforts to develop therapeutics and vaccines and sort of everything else, which is, as you can imagine, been pretty disruptive for a virologist in a virology division who had other plans that didn't really involve a pandemic. Unfortunately, we did have a few plans in place and did have some ideas about how we would respond if this ever happened, and we'll go over some of that today. So talk to me about 45 minutes, and then we'll have some time for discussion. So I did include a background slide or two. A virologist just won't give a talk without telling you about the virus itself. So SARS-CoV-2 or SARS coronavirus 2 is the causative agent of COVID-19. COVID-19 is actually the clinical disease caused by this virus. And this is a member of the coronavirus family, and there's several other ones. The first four here, 229E, NL63, OC43, and HKU1, are sometimes called the endemic coronaviruses. These spread around the world constantly. They cause essentially the common cold, one of the many causes of common cold. Probably second, the rhinoviruses is the cause, but nothing particularly severe about those. But there are bad coronaviruses. Everybody remembers that if we're living through SARS-CoV-2, there is a SARS-CoV-1. This happened quite some time ago now, and had quite a different course. And I won't go into this too much, but quite different from what we've seen in a very severe disease with a very high fatality rate, but also had some really impressive and important differences from SARS-CoV-2, such that it was actually easier to identify people who were infected. They became sick before they actually started to spread the virus. It's actually much easier to contain this virus, and it never became a global pandemic. And then there's MERS, the Middle Eastern Respiratory Syndrome. This comes from camels and jumps into people, and has a very, very high fatality rate, but doesn't seem to spread between people well at all. So something we're keeping our eyes on. But you can see that there's a tremendous spectrum of disease that these viruses can cause. Now, they're positive-stranded, single-stranded RNA viruses. So the RNA, this is important for how we detect them. We'll talk about this. They have actually a big genome for an RNA virus. Typical RNA virus will have no more than about 10,000 base pairs. That'd be something like HIV or hepatitis C. And this is 30,000. Because it's so big, it really can't tolerate very many errors while it replicates. So it actually, uniquely among RNA viruses, has a proofreading exoribonuclease. And what this means is that it actually mutates substantially slower than most RNA viruses. And we'll talk some about mutation. It's very much in the news now with these variants from different parts of the world. But this virus really doesn't mutate that quickly. And then it has certain structural proteins. Envelope will be important for serologic testing we'll talk about. And we do some testing for nucleocapsid as well. All right, so there's the virus. And as of yesterday, this is sort of the hot spots around the country. And you can see we're up here. We're sort of in a light yellow or orange color, which means it's not too bad. Not that long ago, the Dakotas were terrible. They've calmed down quite a bit, but we're still having problems in Texas and other parts of the Southeast. But it's really more dramatic maybe than where we are now, is sort of the cumulative look of everything we've had. So the size of the circles, the number of cases, and remember that the first cases in the United States landed right here where we are. And our circle's not that big. And you look at the overlapping circles for all these counties up here in the Northeast and in other parts of the South and Midwest. You can see that this area, this region is actually done quite well, at least by American standards, in response to the virus. It's something people can really feel good about. And I hope that some of our efforts actually played into that success. So the first thing I'll say is that we knew this was coming, or we knew that this might be coming, because there were the reports in late 2019 about this disease in China. Hey, I'm going to ask folks who are in the background to just go on mute. I'm getting a little bit of feedback here. Thanks, everybody. So we knew it was coming. We were able to design an assay, and I'll talk about that. But one really important point that I want to make early on is that actually, even if the first global case anywhere of SARS-CoV-2 had happened here in Seattle, we would have found it. And that's because we have what we call a metagenomic pipeline. That is, we don't need to know what virus we're looking for in order to actually find a virus. We can essentially sequence all the nucleic acid in a clinical sample and look for any sequences in there that actually look like a virus. And so very early on within the pandemic, once we had some cases, we actually did our metagenomic analysis. And basically, without any prior knowledge of SARS-CoV-2, we could actually put these sequences through our pipeline and actually get out a response that came out as SARS-related coronavirus. And so we would have known we were dealing with something like SARS coronavirus, and turned out in this case to be SARS coronavirus 2. This is really interesting. So here are the very early cases. It's probably a little too small to see. You can see it over here, like, UW case 1, 2. So this was the second case in the United States, actually, UW1. The first one was from public health. But you can see that these early cases we were able to find, and actually some of them had co-infections. So we could also see, for example, one of them had a parainfluenza virus at the same time. It's respiratory virus season, or it was at the time. There were other things. Some people had colds. Turned out they didn't have coronavirus. They had rhinovirus A or C, and some had bacteria and other things. Now, this is really important because it means if something else happens, we're ready to go. I'll tell you, we do a lot of this still. We never see co-infections now because we've pretty much stamped out all the other respiratory virus infections. There's really almost none other than coronavirus right now. So we were sequencing, and we got very early on, we got some SARS coronavirus 2 genomes, and basically we get them all up and we put them in a thing called GSED. We also put them in NCBI. These are publicly available databases where people can get, and you start to see that you can actually put these onto this map and actually see the relationship of the various viruses compared to their ancestral cases back here. So you'd actually see where the new cases are falling. And that actually gave us a lot of an understanding of how the virus was spreading very early on. So we published a couple of papers on this. One was we published a paper in which we actually showed that the predominant, almost all the very early Washington cases actually came from a single clade here. And then there was a later introduction of a second clade, and this was the one that sort of came from the East Coast and eventually ended up actually supplanting everything that had originally come from Asia. The early cases actually went from Washington and the Pacific Northwest, or potentially southern British Columbia, down to California. We collaborated with a group down there to show that actually some of the early cases, if you remember, they were on cruise ships. And it turned out that a couple of folks on those cruise ships actually had viruses that looked an awful lot like the Washington clade, like this clade that turned out to not be the major one in the US later on. And it turned out that a couple of those people had actually traveled from Seattle down there. So we're actually able to track in a very early way how the virus was spreading around the country. In fact, we were able to track how the virus spread from the West to the East Coast. And you could actually see, we collaborated with a group from the East Coast for this, Nathan Gruboff. And what we actually found was that very early on within these Yale isolates, there were a lot of the Washington strains here. So you can see those that are all sort of interspersed with some from Connecticut and this area. But very early on, again, these became supplanted by a big influx of cases from Europe. And this strain essentially died out on the East Coast and was totally replaced. So that's sort of what sequencing can tell us. And we'll kind of touch on sequencing on and off throughout the talk. But the big challenge that we were dealing with was how do we actually provide not a niche product like genomic sequencing, but how do we provide diagnostics for everybody else? And this was a kind of an opinion piece that we published early on to say, there's a bunch of potential challenges we were facing in early 2020. And that is, we've got this huge population of people who are at risk for COVID. Most of them aren't getting tested. A few of them get tested, but they're maybe not getting testing in a reasonable amount of time. And so it means that there's only a small percentage of people who are actually getting actionable results. And you remember this very early on, it was very hard to get a COVID test. Very few places had a test. There were problems with the CDC test we'll talk about. And then, there were wait times of 2 weeks to get a result. Well, you couldn't really do anything with that. And really a lot of that had to do with some really regulatory issues around the CDC. And this has been well documented in the New Yorker and other places about how CDC essentially really wanted to keep the testing kind of within their purview. Early on, all samples had to go there through January. We had started making a test. Once the genomic sequence of the virus came out, we started getting ready. I started telling the folks within our hospital that we could do testing for our patients. And then very quickly opinions were passed down from CDC that all samples suspected of COVID had to go to CDC and you weren't allowed to test them locally. Ultimately, CDC manufactured a test, it's shown here. And you may recall that turned out to be faulty. So that went out to public health laboratories around the country, and it turned out that it didn't work for most of them. Which really meant that we were flying blind through all of February and a bit of March. And so we really were stuck. Having prepared for this, developed a test that performed quite well. We actually couldn't perform that testing until the beginning of March. And so we were, I think, the first academic lab actually in the country who was testing for SARS-CoV-2. And so what happened was that the United States Food and Drug Administration realized that CDC wasn't going to be able to provide all the testing that was needed. And so they actually greatly relaxed some of the regulatory burdens that we were frankly struggling with to meet. We're going to cost millions of dollars to actually meet the requirements. They actually relaxed those so that we could get our test up. And so we got the green light on a Saturday morning, and we were receiving samples the next day. So a lot of work went into how do you do this testing. So the first thing we did was we invented our own test. We also looked at the CDC test. We looked at a test that the World Health Organization had developed. Here it's called the Corman primer and probe sets. And a lot of work went into just studies about how well do these various tests work? Are they accurate? Are they precise? Are they sensitive? Is one better than another? And one thing that we found, and it was corroborated by other folks, is that one of the CDC primers, it wasn't just a manufacturing problem, it just wasn't a good design. And this was a CDCN3 primer, which isn't shown here because it performed so poorly. So we're really able to say, what are the considerations around using these and which ones really perform best. That was very useful. It wasn't that long until then the commercial providers started to make tests. So companies like DiaSorin, Roche made a test on their Cobas instrument. The Gene Expert from Cepheid made a test. Hologic put it on their Panther platform. And so we were able to actually spend some time looking at how those actually performed. And there were differences between these. Some performed better than others. These tests were pretty good. They were all compared to our laboratory developed tests, the University of Washington LDT. And most of these worked well, but you've heard some of the problems. Things like the Abbott iNow test, for example, they'd never be as sensitive as these. And we actually provide some of that early data, actually worked with Abbott to try to get that instrument working as well as it possibly could. And it's actually contributed quite a bit to the response subsequently. A lot of background work goes into things like just what kind of sample type is sample type you can to get these tests. A lot of things to do with calls a nasopharyngeal swab. That's the one some people call them the brain biopsy. They actually go all the way up to the very back of the nose. It's sort of uncomfortable. There's other swabs that don't go quite so far up the nose. How well do those work? What about saliva? What about a throat swab? What about a gargle? And so we spend a lot of time on those. The bottom line is that the sample is a nasopharyngeal swab. Everything else is a bit of a trade-off. You also gain acceptability. People will tend to go back if they don't hit quite so much. And so we're able to kind of put parameters around that. We also, our laboratory's done a lot of work in something called digital PCR. So in PCR, you're basically taking some piece of nucleic acid and you're amplifying it up millions of times until it becomes detectable. Typically, we put a fluorophore in it so that it fluoresces and the instrument can see it. And so sort of one way to quantify is that if you just look at the number of cycles that you need until you get a detectable signal, if you only need a few cycles, there must have been a lot of material, a lot of starting target. If you need a whole lot of cycles, you didn't have much to start with, and you can kind of make a standard curve that way. Digital PCR actually splits the reaction into thousands, tens of thousands of individual reactions, and you actually physically then can count the amount of starting material from the number of PCR positive droplets compared to negative droplets. And this provides an absolute quantification of virus. And this is actually important because, and I'll touch on this throughout the talk a little bit later, we were able to show that knowing how much virus is there is actually very important in terms of understanding how a patient's going to do. People with lots of virus typically don't do as well as people with less virus. We've learned that people with little tiny bits of virus actually aren't infectious. And so the ability to quantitate was very, very important. And the ability to use the digital PCR to get that early on was very important. One question that came up early on was how sensitive is PCR for SARS-CoV-2? There were some reports, mostly out of China, that said that there were a lot of false negatives with PCR. This would be very worrisome. If somebody comes in for this test, you tell them they don't have COVID. Well, if it turns out they do, maybe they're exposing other people, not taking the right precautions. slow to go back to the hospital if their condition worsens. So a lot of bad things can happen. And so we started to study basically to look at people who'd come into the University of Washington with symptoms that look like COVID, and then got a negative result. And then how many of those got a second COVID test within a week? So the idea was, if these were people who got a false negative, but they're still sick, maybe they've come back. If they come back, do they have COVID now? And once we started to do that, we actually started to talk with some colleagues at Stanford who had actually trained here with us. And so the director of the Stanford lab was one of our UW alums, and he was actually had designed almost exactly the same study. And so we put them together in this publication. And basically found almost identical data between UW in blue and Stanford here in sort of this tan color. And the bottom line is, if you look at people who've come back for testing within that week, and this is thousands of people, very, very few people who were negative on the first test actually had a positive on a later test. It was about 2.5%. So that's sort of an upper bound for false positives. And we've subsequently learned that very early on in the course of the disease, you don't necessarily have much virus to actually detect. And so some people can actually have a negative test, and the virus shows up a little bit later. So we learned that, but it doesn't mean the test isn't performing well. It just gives you a sense about how confident you can be in it. And the answer to that is very confident. So we had this testing. We had really testing in a big way. We were performing hundreds of tests a day and subsequently thousands of tests a day. And really early on, we were able to really flatten the curve. Remember, the idea was we didn't want to have everybody end up in the hospital at the same time because the hospital just couldn't handle it. And so we were able to do that. So this is just the positivity rate over time, very early on from when we first started, kind of plotted by day through the month of March. You see we reached this peak in Washington State patients, Seattle outpatients or emergency department patients, all right here, right around the end of March. And then we were able to sort of turn the tide on the virus, and the positivity rates went down, back down below 5%. We actually extrapolated these out. Mathematically, the reviewers hated it, but turned out to be actually very accurate with what happened over the next month and a half or so, until we had subsequent peaks later on. We've done a lot of collaborative work through this, and it's been really fun to collaborate with people, clinicians around the hospital, clinicians around the country, bioengineers, other folks. It's been really fun. But one really important paper that we published early on was contributed to this case series, simply asking, here where the virus really started in the US, what is the clinical experience? And the clinical experience early on in this region was really quite poor. Here's a bunch of the early patients, and you can see that a substantial fraction of them actually died of the infection. Part of this was it was really the sickest people who were able to be insistent enough to actually get testing early on. Remember, this was before we were really providing testing for everybody around the state, but the sickest people around the state would come here. And so this is a bit self-selected, but still we were looking at about half of the patients who were actually admitted into UW Hospital early on died of this infection. Subsequently, it's become a lot better, and in the mortality rate in the US continues to hover around 2% of diagnosed infections. The overall mortality rate is probably lower than that, given that there are undiagnosed infections that are quite mild. But a lot has happened. The clinicians, the ICU docs, the nursing staff, they've all learned a lot about how to handle these patients. We have antivirals. We have drugs like dexamethasone that can tamp down the overexuberant immune response that causes a lot of the symptoms that people have late in the disease. Simple things like pronating patients, so having them lie on their stomach sometimes to help breathe. All these things have really helped reduce the mortality rate. Now, one thing I mentioned when I was talking about the ability to quantitate virus is that it turns out that how much virus you have when you present is actually, when you present to the doctor, is really important for saying how you're going to do. So let's look at this left panel. So on the x-axis, we have something we call the cycle threshold. This is the number of PCR cycles that you need in order to get a detectable signal. So if you don't need many PCR cycles, you have a lot of virus. So these down here are people with lots of virus. These are people with relatively little virus when they present. And then plotted on the y-axis is just how long was their hospital stay. And you can see there's sort of what looks like a bit of a kind of a bell-shaped curve here. But now notice here that the X's are actually people who are deceased. And you can see that people with very, very high viral loads were much more likely to actually die in the hospital compared to people with lower viral loads. And that's continued and been corroborated by researchers across the country now. So quantitation can be very, very important. We've also looked at how much antibody they have, and we'll talk about that in a minute. And antibody can also be an important prognostic indicator. So pretty early on, we were asked whether we thought it made sense to actually begin to universally screen UW medicine patients. There was some debate around this. The idea was, well, it's pretty rare that somebody's actually coming into the hospital with undiagnosed COVID. You're going to do a lot of tests. They're going to be negative to find very few positives. So we tried this originally in the maternal fetal medicine clinic, where there was universal screening. And we actually found, out of 230, we actually found a fair number of positive cases, a total of 5 here, 4 were symptomatic, 1 was asymptomatic. But in fact, if you look at that, and you start to think about, it seems like a low rate, but you think about the ramifications of having someone come in with undiagnosed COVID, being in a general medicine floor, having full contact with others, there's a tremendous risk for within hospital spread. So this actually became a standard of care throughout the hospital. In fact, right now, patients who are in the hospital actually get periodic screening, so not just at admission, but actually throughout their stay, just to try to make sure that we don't have these outbreaks within the hospital. We've also did this within people who are getting aerosol generating procedures and got pretty much the same sort of positivity rates, about 0.5%. We spent a lot of time looking at what's going on among our own healthcare workers. This is just some of the early data. We were obviously very worried about our staff getting sick. First of all, we owe it to our staff to keep them as safe as possible. And second of all, if the healthcare workers all get sick with COVID, they can't take care of anybody. So there were a lot of personal protective equipment shortages early on, a lot of scrambling to be able to find things to keep the staff safe. But in fact, what we found was that if you actually looked at the prevalence of infection, either among our frontline staff here or our non-frontline staff here, it was actually almost identical, which meant that these were the people actually who were dealing directly with people with COVID. These are people whose most of their exposure was within the community. And they were essentially the same, or slightly below the overall rate within the community. So what this really meant was we were keeping our employees very, very healthy. And that was a lot of reassurance to the hospital infection control folks that they were doing a good job. One of the stories that I'm actually really proud of, so we developed this ability to do this testing, and I mentioned we're doing hundreds and then thousands of tests. We were very proud of the fact that we could not only do lots of tests, we could actually do them very rapidly. So if you, out of all your choices of places to go and get a test done, if you actually went to UW Medicine or to one of our client clinics where the testing came to us, we've really never had more than about a 48-hour turnaround time from sample receipt to result. Right now, it's about 10 hours from the time we get the sample till a result goes out on average. And what this rapid turnaround allowed us to do was to think about other parts of the healthcare system that were tremendously impacted by COVID. And so one of those was the solid organ transplant programs. And so basically, if you look at sort of the different parts in the country, so the Northwest, National, Southwest, Northeast, essentially, these were the percentage of people who were inactivated because of COVID. So basically, they shut down the transplants. Okay, you can't get a transplant because there are no organs, or we don't know that it's safe. And essentially, very early on, we were able to provide this rapid testing of both prospective organ donors as well as recipients. And we were able to actually get this reopened so that within a week, we were actually able to drop the proportion of people who had to be put on the wait list very, very low. And so that was able to continue on. And a lot of people got their transplants because we were able to do that. So we were very, very pleased about that. We spent a lot of effort looking at congregate living facilities. You might recall that a lot of the early outbreaks in our area were within nursing homes, assisted living facilities, and so forth. And we worked with some of our public health folks like Allison Roxby and Santiago Nimi to work with some of the local facilities and see whether intensive screening of staff, residents, and potential visitors could keep COVID from actually just running rampant through these facilities like we'd seen in some of them early in the pandemic. In fact, we did this, and this is some of our examples, is that by doing this, we were able actually to identify residents and a couple of staff members who had essentially asymptomatic disease, get those folks isolated from others. So they're shedding virus, but they're not showing symptoms, at least yet, or maybe ever, but they could infect others. So we're able to get them separated, and we had no outbreaks among these facilities. And this has become really a model for facilities around the country. The state of Washington has put in mandatory periodic testing in these facilities, and there's been much, much less problem in these settings than there would have been otherwise. So we've been doing a lot. This is our cumulative testing since the very beginning. Here's the date on the x-axis, y-axis is how many tests did we do that day, and little spot down here in gray is what proportion of those are positive. So you can see here's sort of this initial, we get the green light. I think the first day we did about 20 tests, and then we did 100, and up it goes. And you can see this first peak, and we had sort of quiet time in the spring. We had a summer peak testing, and then you can see this big autumn peak as well that we are hopefully on the back end of now. So we've done almost 2 million tests. This is up to date as of yesterday, I think, or maybe the day before. Most recently, our most recent positivity rate is 4.7%. So that's down quite a bit from where it was. Here at the peak, we were up about 8, 9, 10% on most days. So we're going in the right direction. We've done an awful lot of tests, and we're not done. And we're going to continue to do this. We'll pass 2 million probably later this week. And depending on how the spring and summer go, we may end up doing 3 million tests. Shift gears a little bit from PCR and talk a little bit about serologic testing. So PCR is we're actually looking for the virus infection itself. So we're detecting the viral genome through this polymerase chain reaction. But you can also look in the blood for a marker that the body's been exposed. If the body gets exposed to pathogen, it'll typically make an immune response, and part of that is making antibody. This molecule looks kind of like this, and kind of up here at the very tips is the part that'll actually bind to the virus itself and hopefully prevent it from infecting new cells. But you can take the blood and look for these things. And then you can get a sense about within a population, how many people have been infected. Maybe that'll help policymakers. You can tell people maybe something about how much are at risk, and then can we go back to work, and these sorts of things. So we thought there'd be a lot of utility in the serologic test, but there's really certain things you want in a serologic test, and these are not a given. So you want it to be very sensitive and very specific. Now, those have very precise definitions within my field. I'll kind of paraphrase them a little bit. So sensitivity is basically, think of it as the likelihood that if someone actually has past infection, that you're able to detect it. So, what percentage do you actually detect? Specificity is if you say that somebody has been exposed to it, they have these antibodies, what's the likelihood you're right? In other words, do you have any false positives with this? This was very, very important early on because among the early antibody tests, they had a lot of false positives. So You were telling people they'd been exposed to COVID when they really hadn't been. People thought they could, not worry about it anymore. They'd already had the infection. We got some bad estimates of how many people had it in the community. So specificity was very, very important. We'd like to know that the presence of these antibodies actually means something. So does it actually have some protection or something? And then we'd like to be able to do a bunch of them and ideally put them on instruments we have. So we identified an assay that was in development from a manufacturer called Abbott. This is the Sarco V2 Igg Assay. And it essentially uses this sort of chemiluminescence microparticle immunoassay. It's the actual chemistry that detects these antibodies. It detects antibodies that bind this nucleocapsid protein of SARS-CoV-2. And I didn't go into it too much at the very beginning, but the coronavirus has those spikes that come off of it that make it look a little bit like a crown. And after infection, you make antibodies to those spikes. Now, you also make antibodies to something called nucleocapsid. Nucleocapsid is actually on the inside. It wraps up the RNA, the actual genome of the virus is wrapped up in these proteins. And so it takes a lot of this protein to wrap the entire genome. And so you have a lot of it, and therefore there's a very strong immune response to it. So it's very, very good for detecting exposure. I'll say that having an antibody to nucleocapsid won't prevent the virus from infecting a new cell. So it doesn't do something we call neutralization. And for that, you need to actually measure antibody to the spike. The spike is what allows the virus to get into a new cell. So if the antibodies bind that spike, they have a fighting chance of actually preventing the virus from getting into a new cell. So you can make the diagnosis here, but it's not completely clear that it correlates with protection. Well, I'll show you a little bit of data about that in a minute. It works on this immunoanalyzer that we have. There's tons of them around the country, and each one can do about 3,000. So a lot of ability to perform this testing. So we got the first look at this around the country. The manufacturer had actually put out some of their initial data on it, but we were able to independently take a look at that. And so we looked at 125 hospitalized patients within our system who had tested positive for SARS-CoV-2, for the virus itself. And then we just plotted something called an index value. So this is, you can think about, remember it's a chemiluminescence assay. So it's how much luminescence there is above some sort of background level within the assay itself. And essentially anything above this 1.4 line will be considered positive, okay? So anything above that red line we'll consider positive. So if we look at days since symptom onset, remember, these are people who we know have COVID from our PCR testing. You can see some of them are negative early on. A few are positive right at the very beginning, but then over time, more and more of them become positive at higher and higher index values. You can also do this plotted not since symptom onset, but since PCR positivity. You get the same sort of curve. It's just shifted over a little bit. You can see that people become PCR positive before they have symptoms. And that's why this is a really hard disease to contain. But you see that sensitivity actually goes up. So this is what percentage of these people who have this disease we actually detect. And actually by 17 days after symptom onset or PCR positivity, everybody has antibodies. So it's 100% sensitive, and that's a really nice performance in that regard. It's also highly specific. We were fortunate enough, because we're a big virology program, we have big banks of sera that's been sent to us for other things. We happen to have over 1,000 samples that were sent to us for herpes simplex virus diagnosis in the 2 years before there was COVID. And so we tested all those, and of 1,020, exactly one would have been determined positive. I remember this threshold is 1.40. This one came out at 1.41, so very, very borderline. And that is really, really great performance. So compared to all the other assays that existed early on here, this was a great one. Sometimes we present this as something called a receiver operating characteristic curve. So basically you can plot the sensitivity versus specificity. And so kind of a worthless test, goes from no sensitivity to but perfectly specific, so it really doesn't tell you any information. A perfect test looks something like this. Most tests have a curve like this, and you're always trading off sensitivity versus specificity. But here, if we made our own cutoff at 1.42 to 1.49, we just have to get a perfect ROC curve. And that's something I've actually never seen before in my career, a perfect assay. Um, Another really cool thing about this is that we could actually follow those people who were negative when they first came in for antibody. So we know the virus, but they have antibody yet. And you can actually follow them and watch them become positive during their hospitalization. So each of these curves is an individual person's index values going up during their hospitalization. You can see it happens actually quite acutely within a day or two. Suddenly they've got a lot of antibody. And the earlier you have antibody compared to when you're sick, the better off you are. So we've done a lot of studies looking just at seropositivity in different parts of the country. One very early on we did in Idaho. This was in late April. About 2% of people in this study had antibodies. We tested a lot of people at EW Virology. This is up to date. This is actually pretty out of date now. This would have been as of last summer. About 5% of EW Virology. All the samples we tested were positive. Just all comers. We've looked at our own, our own employees. That's going on. This is a pretty low number. It still stays, I think, around 3%. So it's actually quite low. But these are healthcare workers who know how to keep themselves healthy and so forth. So we really would like to know, from a point of view for statewide policy making, to have a better sense around what's going on on a statewide basis, particularly now as we're starting to have vaccination coming through in addition to natural infection. And so we've actually teamed up with the Paul G. Allen Family Foundation to do a statewide seroprevalence study. So we'll be looking at 8,000 participants, and everybody's getting PCR, and then the serologic testing over a period of months with kind of follow-on testing at a couple of month intervals. And this will be really a random look across the state. We'll be doing testing that can actually distinguish infection, actual infection with COVID versus the vaccine. If you get the vaccine, you have antibodies to the spike, but not the nucleocapsid. If you get infection, you have antibodies to both. So we actually tell those apart. And we're hoping to learn a lot, not only about just where has COVID been around the state, but also things about the duration of protection, and then a better understanding about protection from vaccination or natural infection versus the viruses we have now, as well as the variants that are circulating and likely to come here in the future. Okay, so one of the things I mentioned, we have this nucleoprotein, or this nucleocapsid, ELISA. We're detecting antibodies for this component that's on the inside of the virus. It can't neutralize. How well does it actually correlate? And do antibodies actually protect at all? So that was a big question for months. And we were lucky enough, actually, and Alex Greninger from our group really led this. It was a good insight on his part. There was actually the first demonstration that antibodies actually protect you. And how this worked was, UW Medicine had been consulting with the fishing industry, how to keep these fishing vessels safe. Remember, there have been all these outbreaks on the cruise ships because of the tight quarters and so forth. Well, that's magnified many times on a working fishing vessel, where people are working long hours, frantically, very crowded conditions. And so there was a lot of interest in testing crews before they went onto the vessels, so that we knew that nobody was carrying the virus onto the vessel and it could go about its business through the fishing season. Of course, a few people actually didn't follow all the recommendations and cheated and so forth. And so one of the vessels actually did have an outbreak. And then we were actually able to go back to the samples that we had gotten from before the vessel left, and actually look whether anybody had pre-existing antibody. And the answer was that of this crew of about 120 folks, 3 people had neutralizing antibodies before they even started. So they'd already had COVID and generated a very protective immune response. None of them got infected, where almost everybody who didn't have those antibodies got infected on this boat. And that was highly statistically significant, even though it was only 3 people who had those antibodies. And that was actually the first demonstration of this. It's been demonstrated multiple times now. That the antibodies protect. It turns out that you can actually then look at sort of this nucleoprotein ELISA and compare it to much more sophisticated but difficult to perform assays and see how well they actually correlate. And the answer is that if you have these nucleoprotein antibodies, it's a pretty good predictor that you probably have protection. It's not perfect, and you can have these without having neutralized antibodies, but it's pretty useful. So the darker red is the better the correlation. So it's sort of one-to-one thing. So there's a few red and certainly all some pink in this, compared to something like a neutralization assay here. So definitely tells you that there is some protection. Just to wrap up in the last 5 minutes or so, we've been doing a lot to try to speed up testing. There seems to be an insatiable demand for testing. You can see we were some days doing up to 14, 15,000 a day. So we've been pooling samples. We were the first lab in the country to actually be pooling samples. And so that was really pretty cool. You basically take, if you have samples from somebody from a population is pretty unlikely to have COVID, you can mix. In our case, we mix 6 of them together and test that entire pool at once. If that's negative, you tell everybody you're negative. If that pool's positive, you pull them all out and test them one at a time. Saves a lot of work in reagents, allows us to do more testing. It's worked very, very well. We've also done things like try to take steps out of the whole process. So the beginning of this, you do something called a nucleic acid extraction. We actually pull the nucleic acid out of the virus. We've got lots of sophisticated instruments to do that. And once you have it pulled out of the virus, you can do this PCR, this reverse transcription and the PCR reaction. But we actually found you could take out that extraction. You lose a little bit in sensitivity, and that's just this jump here between these 2 lines and how many cycles you need. But the testing still performs quite well. Really important and very useful in parts of the world that don't have access to these sophisticated instruments that we do. And so this has been actually put into place in laboratories around the country, including the Children's Hospital in Philadelphia. I was surprised. They're actually doing this clinically routinely, but a lot of third world and developing countries have been doing this. We actually have done a trial where we looked just to make sure this really works in real world. So we worked with labs around the US, in the Caribbean, Europe, South America, Africa. And the answer was, it works really well. Everybody gets pretty much the same answer. And I won't go into this too much, but these are sort of residuals. How many cycles are they different? And this is really a trivial difference in the test, and really doesn't matter at all. It really works well. The positive and negative agreement was actually perfect between the labs. We've looked at approaches that don't take PCR. PCR is just one way to amplify nucleic acid. You want to take this little bit of virus and amplify a fraction of it into a lot of that little fraction so you can actually detect it. So you can do things that don't require the same instruments and the same reagents. This is good because it just gives you ways to get around equipment and supply issues. So we looked at something called loop-mediated isothermal amplification. That worked pretty well. Kind of those of you who are familiar with the CRISPR system, this was really neat. These isothermal systems, like the lamp that I just showed you, have often had a trouble that they have some false positives. The stuff starts to amplify, but it's not really what you wanted. You get a signal, and you don't realize that that signal isn't what you're trying to detect. So these CRISPR-based methods put in a second step where you get the same amplification of RNA, but then this CRISPR, either Cas12 or 13, actually has to recognize this. And if it does recognize that, yeah, it's the right thing, then it cleaves the substrate and makes a signal you can actually detect. So we worked with actually one of the really co-inventors of the CRISPR system, in order to bring this in clinical testing, and that is actually commercially available now. Same thing there. You've heard some about antigen testing. So, the PCR test costs probably about $100 each. That's what the reimbursement is for them. You can also do antigen testing. It's a little bit like a pregnancy test. You're not now looking for the nucleic acid, but you're using that same technology a pregnancy test might do to look for the protein in the virus. So it's just this little card, takes about 5 minutes. That costs about $5. And it is certainly a reasonably performing test. It has some false positives. It is about 1,000-fold less sensitive than our PCR. These are just some studies we did, look at how sensitive it is. Far, far less sensitive than PCR, but it does detect the people who are most likely to be infectious and infect others. So there's a trade-off between this great sensitivity, but at cost and a cost in money and time, versus a test that's not quite as sensitive or specific, but it's very, very quick, and you can think about doing it day after day after day. So some good trade-offs, and we're seeing more and more of these come out in settings across the country. So just to wrap up, I think that we're likely to continue to see more and more demand for PCR testing, for other kinds of direct testing, these antigen tests and so forth. Unfortunately, as future waves of infection come, as we try to open up the economy, maybe we'll have these variants, and we may have waves because of those coming forward. I continue to think there's going to be more and more demand for serology. We know now that the positivity on serology actually correlates with some degree of protection from disease. We understand the nuances around that, but there are always complications. How well does the serology, as we're measuring it now, protect from the new variants that are emerging? And that's a topic we're looking at right now. We've been helping with development of therapeutics. Didn't talk much about that. We were involved in a study, for example, of hydroxychloroquine that showed that it didn't work. I don't think I even showed that data at all. It wasn't a useful therapeutic. We've been involved in some remdesivir studies. Not a ton of things out there that seem to be particularly useful. The real problem is people get diagnosed late, and then there's this huge immunopathological component. It's really the body's reaction as much as the virus itself that's causing the disease. But we have been supporting the vaccine effort. It has really gone well. As you know, there are a number of vaccines available. About 10% of the population now has gotten at least one vaccine. We're supporting one of the vaccine trials as the main laboratory, as well as doing sequencing for a number of them for the studies about how well the vaccine works against the new variants. So I hope that everybody can get access to a vaccine very, very soon. With that, I'll wrap up just by thanking. I tried to put up names as we went. It's all very fast, but lots of collaborators at Fred Hutch, UW Virology, throughout the Department of Laboratory Medicine, Pathology, all of UW Medicine, and again, just everybody who collaborated on this. I'm very, very grateful. So with that, I'll stop, and I'm happy to take any questions.
Awesome. Thank you so much. That was a really wonderful talk. All right, we can now move into the Q&A portion here. Just a reminder to everybody, if you do have questions, you can either put them into the chat and I will read them off, or you can use the raise hand function in your participants tab, and I can call on you, and then you can unmute and turn on your camera and ask your question. And I guess I have a question to get us started here. I'm just curious, you mentioned kind of the new variants of the virus that have been popping up, and I'm just curious, I don't see any reason there would be, but is there anything about those new variants that change or that would affect the tests that we're currently using, or like would we have to change anything about our testing methods?
Yeah, that's a really important question. So when you do these PCR or other based assays, you essentially you're amplifying a little tiny fragment of the virus. You don't amplify the whole thing, so it's not infectious. But if any of those mutations happen to be right in that segment that you're amplifying, it can change whether the reaction will work. So we keep a really close eye on that. Every time a new one's identified, we look at our laboratory developed test. Will that work? The manufacturers do that as well. For one of the manufacturers, it turned out for the B117 variant, and that's the one that was identified in the United Kingdom, their particular assay from this particular manufacturer had it amplified two regions of the virus. One of them was unaffected by the mutations. One just didn't work at all because of these mutations. And that turned out to be really actually very useful, because if you had a positive reaction in one but not the other, that was a pretty good indication it was probably this B117 variant, and you could go on and confirm that by sequencing or something else. You clearly don't want to have a situation where they both fail. So most of the assays, not all of them, but most will interrogate two regions, just so that you have a chance to fix things before it completely starts to fail.
Great, thank you. Okay, looks like we have our first question in the chat here. This is from Tina forgive my pronunciation, is it Chimes or Chimes? Are there any research studies ongoing that suggest how long the antibodies provide protection against reinfection, or is it too soon to have this information?
Yeah, well, that's something that seems to be uh, it's an ongoing question. It's incredibly important. So early on, most of our guesses were based on experience with the other coronaviruses, and if you look at that, it seems that if you get one of those endemic coronaviruses, you're sort of protected from that same coronavirus for a year or two, and then people could kind of get reinfected. Um, and so people extrapolate from that that SARS-CoV-2 hopefully would be something like that. Then there was sort of this flurry of reports of people getting infected twice, these reinfections. Remember that? Somebody got really worried that there wasn't any protection. It seems like those are sort of fringe cases. People had a very mild case, didn't make much, if any, antibody, and then actually got a full blown infection the second time. Um, so what we can say is it looks like the antibodies last at least for a year, or close to a year, at least in kind of most people. They're still detectable, and that's because that's sort of as long as we've been able to look. So I think some people definitely lose antibodies quickly, and so the antibodies begin to decay once you clear the infection, but for many people, they remain detectable for a year or more. It looks like people who get the vaccine actually get much better antibody responses than people who get the infection itself. And so there's some hope that those antibodies will stay even longer and you'll get a couple of years of protection. But the reality is it just hasn't been enough time to really know for sure. Um, we do see that people who have very mild infections can lose their antibodies very quickly, and some of those they really do become undetectable in a couple of months. So it's variable, but I think it's generally more good news than bad on this.
Great. Okay, I'm going to answer ask Rory McGuire's question in the chat, and then I will call on David with the hand raise. So the question from the chat is, I heard about some really cool test pooling methods developed in Rwanda earlier in the pandemic based on hypercube geometry, and there's a linked Nature article about that. I've been curious why the methodology hasn't been adopted more outside of East and South Africa, since it seems to be more cost and time efficient than standard pooling methods.
Yeah, so I don't know if we're talking about the same paper or not. There's also one that I know was co-authored by a colleague who was at Fred Hutch for a while, is now in Israel, who had a more sophisticated pooling method. Yeah, those exist. They the choice of pooling methods, actually, the optimal pooling method is actually a function of your positivity rate as well. So it's not just there is always one best pooling method. So the simpler pooling methods, just the straight up, 6-to-1, and then go back and pull them out, very, very simple, basic pooling thing, actually works quite well if you have low rates of positivity. It's very, very simple to program instruments for this. It's difficult to mess up because you get 2 looks at each positive. So you test it once, and you go back and you test it again. So you're very, very confident in that. Typically, these other approaches have some sort of matrix by which, depending on exactly which pool something comes up, you can extrapolate. Usually, each individual sample appears in multiple pools, so that knowing exactly which pools, you can deduce which sample that had to have been, but you don't actually go back and test those. And so that's a couple of things. Those are harder to set up front. They tend to be more error prone when you're doing literally thousands of them. And we've never had the positivity rates where we have felt like we had to go to that. So we've looked at that, haven't done it. I don't know a lot of clinical labs who have, but they're very, very clever approaches, and it's a great thing to have in case we get to the point we need it.
Great. David, if you want to unmute and ask your question, go ahead.
Yeah, thanks for the really interesting talk, Dr. Jerome. I was curious what the progress for creating diagnostic tests that work at the point of care or like at home, what progress is being made there right now?
Yeah, probably the best one for at-home use would be that little antigen card that I showed you. They're variants of that. They're very simple to use. I mean, there's sort of no reason that they literally are like these pregnancy tests, or they're at-home HIV tests that work on the same principle. They're lateral flow assays, basically. Um, so it's hard to mess them up too much. They'll often come with some sort of app where you actually read them on your cell phones. You actually take a picture, and you don't have to look at it and guess, is that positive or not. So those things are all pretty cool. Any sort of molecular test typically requires some sort of instrumentation, and that starts to get a bit expensive for at-home use. If it's sort of like if people have chronic condition, you might want to take your blood pressure every day, or you want to check your blood glucose something like this. You can buy an instrument that costs $100 or whatever and do that. Will somebody want to do that to have a miniature PCR, or lamp instrument? Even lamp at least needs to maintain a constant temperature. So, there is some cost into that. So I think probably it's going to be the antigen tests. The reality is those tests, there was a lot of interest in those tests, their promise, because the performance it's not quite as good. You have to be very, very careful about how you use those. And there's actually disagreement in my field. I tend to be a person who thinks they're pretty useful. I think my opinion is actually a bit of a minority viewpoint, frankly, among laboratory folks who really want to just use that kind of, gold standard, near perfect test. A less perfect test, if you can do it frequently enough, can actually make a difference. But if you use it wrong, you can cause a lot of harm. I'll give you an example of that. So early on, people wanted to use these antigen tests in nursing homes, great, but they wanted to use them to identify which people should go into the COVID side of the facility versus the not COVID. Well, if they have false positives of about 2 or 3%, by the time you do 100 of those, you've sent 3 people who didn't have COVID over to the COVID side, right? We're going to get it, or vice versa. So, it can be useful there, but you got to have a protocol where all the positives get confirmed by, so you can't use them wrong. That's the thing.
Yeah, that's really interesting. Thanks.
All right, going back to questions in the chat, and we have run up to 2 o'clock here. So before we move on, I just want to say thank you everyone for coming. Thank you again, Dr. Jerome. We really appreciate your time. And if you have a couple of minutes, I think we'd love to get through these last couple of questions.
Yeah, I can stay about 5 more minutes. Yeah.
Awesome. So this one is from Evan Pepper. He asks, have sequencing errors and bad bioinformatic pipelines / genome assemblies been problematic in terms of identifying true mutations and new virus variants?
Well, I'm sure there have been those. There's a lot of sequence out there. There's hundreds of thousands, if not millions, of sequences now. So I'm sure that there's some garbage out there. It's not many major applications. If you see a virus that appears one time, it's sort of a novelty. It's when you start to see that appear multiple times, and you won't make the mistake multiple times. So it's had too much trouble yet, but there's really no clinical utility.
Uh-oh, I think we may have lost him. We'll see if he comes back here in a couple of seconds. Otherwise, it sounds like he's got something else to be moving on to.
I don't think he's coming back.
Yep, it looks like we may have lost him. So thank you again, everybody, for joining us today. A reminder that our next presentation in the series is on the 16th with MolES professor and chemical engineering professor James Carruthers. So we hope that you will be able to join us then, and have a wonderful afternoon.
No, I'm back. Sorry.
Not a problem. So I think,
Lost my signal.
yep, no worries. And I think we can let you go here since it sounds like you've got something else to be getting to.
All right, good.
Thank you so much. We really, really appreciate it.
All right, take care.
Yep, you too.